So all that's the biochemistry that you can do if you want to stress bake or something like that. And that reminds me of Christmas. There's actually a chapter 10 and 11 type topic that reminds me of Christmas that I think is really cool. So remember that liposomes are just basically big versions of vesicles that you can make, and they form naturally if you just put fatty acids in water. It's something that has a hydrophobic and hydrophilic part. And if they're long and skinny, then they will come together into membrane-like things. The interesting thing that happens is what happens if you mix two of these together. That they're compatible, they have compatible tails, but maybe they have different chemistries on the head groups. They will actually form some kind of phase separation that will happen. Depending on what you do, you can label one red and one green, and you can see these different patterns that result. These look like living cells. All they are is two different kinds of lipids, probably fatty acids of some sort. I don't think they're even complex enough to be the membrane lipids, but they might be. Actually, I think some of them are. So these are basically just membranes, though. They're simple membranes because they have two components. You mix them together, and they automatically make those cool shapes. And those look like little Christmas ornaments, you know. They're really tiny Christmas ornaments. But I've always wondered about if you take a picture of this and hang it on your tree and tell everyone it's Christmas, it's a Christmas ornament, they'd believe you, right? So the coolest thing is if you take ceramide, cholesterol, and glycerophospholipid, this is why I know that these are triacylglycerides or something like a membrane lipid, because that's what ceramide and that's what glycerophospholipid is. This is a relatively simple composition, but it makes this star. So you literally can make a Christmas star ornament just by mixing three of the right lipids together. This domain, for whatever reason, results in a 5-pointed symmetry like that. And it's just phase separation, like the lipid rafts that we were talking about, how certain lipids will self-associate, but it's all still liquid. So these are liquid flowing sort of structures. This is a paper about it, and this is what I want to show you what this paper is. The important thing about this paper is that it's really pretty simple. You're mixing a couple of lipids together, and you have it in a buffered salt solution. And then you can take them from one solution and put them in another solution, where the only difference is salt. No ATP, no ATPase, no proteins around at all, in fact. This is entirely chapter 10 and 11. But what happens if you take this and you just transfer it from one salt solution to another, you get something that really looks alive. It's kind of like the counterpart to the cornstarch movie that we showed, where the cornstarch looks alive if you put energy into it and just let it shake and then disturb it a little bit. Here's where you're putting energy into it in a sense by transferring it from a high salt solution to a low salt solution. It has salt in the middle, and then you have osmotic pressure across the membrane that will be the driving force. Just based on that simple driving force, I want you to see what happens. For example, if you take it if you form it in an isotonic solution, then you take it to a hypotonic solution, you will start to see like microdomains forming, and then they'll like build up, and then it will like release some of the osmotic pressure, and then they'll disperse. And you'll end up with like a cycle where you have these shapes forming, polka dots, forming and unforming. Again, this is just taking liposomes and moving them from salt to another. Now, after a while, they run out of gas. The osmotic pressure difference, like it will push on it and then it will pop out, and then it will push on it again and pop out. So it'll go through the cycle like 4 or 5 times. So this definitely runs out of gas after a while because it will equilibrate. First chapter concept, homeostasis and equilibrium. But when it's in a case where you have a bigger difference or you're farther from equilibrium, you have something that looks very lifelike. And it's just driven by membrane tension. It's driven by osmotic pressure. So what I want to show you, one is what I want to show you. This is just what happens, and this is just from Brownian motion. This is just from having the atoms going around to each other. But even this looks alive. A simple mixture of lipids under normal conditions looks alive. And it gives you an idea of the scale there, the 10 micrometers. So 3, 7, and 8. Here's where you have a bunch of them together. And this is where you put them into a hypotonic bath. And you can see they're going through those polka dot cycles. Phase separations, and then the pressure is relieved. But the pressure causes the phase separation in some kind of weird kind of thing. So lipid rafts aren't like something that's arranged by the cell. They're something that sort of happens spontaneously. And I'm just interested in how these are so lifelike. Was 5 one of the ones? Oh, I keep forgetting. 7 and 8. Okay. So you can see other ones. All right, here is when you have 2 next to each other. And so sometimes the phase separation can be so much that you actually have little pores forming. Remember, electroporation forms little pores by putting electrical stress. This salt stress can result in little pores forming where it opens itself up and then snaps back together. You know, like it relieves the stress and then it snaps back together. So I just think that's really kind of amazing. And again, that looks lifelike. It looks like something that is forming pores deliberately, but that's a consequence of the two. And here's one interesting thing. Here's the miracle of birth. Did you see that one just sort of popping out? You can actually have one bubble divide into 2. And then you have the 2 that are sort of the mother and daughter cells there. That sort of, you know, let's watch that again because it happens right away. Do you see that at the beginning? I could watch that repeatedly. So it actually starts inside and then pops out. So you can have things like that. That's very lifelike behavior, and it's really interesting to me that it's just lipids. So phase separation is actually a big area of study now. They're studying how cells probably use the fact that proteins form these sort of self-associations. And it's possible that this phase separation inside the cell led to organelles of some sorts. Membranes also form around those, so there's a lot of lipid chemistry going on there. Okay? And in fact, here's a paper that just came out about how it can explain how pollen forms. Now again, this is like bread. We don't know exactly what the structures of each of these are, but we know that there's a lot of saccharides in each of them. So the way that pollen forms, a cell divides, but the mother cell wall basically starts and stops to divide. So you have a regular cell in green inside the mother's cell wall in blue. And so you have some kind of polysaccharide. It's a bit like that last video. It's just what would happen if the cell started to erupt, but then got a cell wall. We have the complication of having a cell wall and a cell membrane, by the way. So, but the idea is that you're encased in a cell wall. It's blue. And it's some kind of polysaccharide. It has a lot of weird beta 1 to 3 linkages. From your study of sugars, you should know beta 1 to 3 linkages are unusual. Part of the thing that makes them tough and harder to break because they don't have as many, there's not many enzymes out there to break them. But they're also nice, tough glycosidic bonds. So you have the callose on the outside. Then the daughter cells start to secrete this purple layer. It's called primexine. And we think it's mostly a polysaccharide because we know that it has a lot of sugars in it. We also know it flows like a membrane. So it's kind of like if you can imagine a bunch of polysaccharides that are like the lipids, and they're able to flow around each other in a sense. Okay, and again, it's like we saw that with bread, and now we're seeing it with polysaccharides. The important thing is because they can flow, because they're fluid, you can actually have some versions that are like to self-associate. And you end up with some self-associating. You see the dark purple and the light purple. Some of them gather together, and that starts to press on the membrane, and it starts to make these patterns that look like those lipid domain patterns that we showed you. It's not lipids, it's saccharides. So this is how they associate each of these with different stages of pollen formation. And what they're saying is that these look like they're domains. They're like lipid domains, but they're sugar domains where the sugar has self-associated. And then the cell makes different parts. At the thin part, it starts to make different proteins and other structures. But the reason why they're so symmetric and they're so kind of they're like the domains that we showed you is because there is actually a phase separation going on. This is where they show a bunch of different pollen spores. And you notice how all of these, they do look like those membrane phase separations that we were doing. So I think that, I believe them. I think it's kind of a cool theory for how do these cool patterns form. By the way, on the left are actual pictures of pollen. On the right they used their computer simulation to build up these pollen patterns from just minor differences in the composition of the different layers that they're talking about here. So it looks like from a minor difference in chemistry, you can get major differences in how things look, but they're always symmetric. I mean, it's like, this looks a little bit like that star that we showed you. And so I think there is a deep similarity, and it comes from self-association, okay? You don't have to have a protein like laying these down like bricks. They come together by themselves. And to me, that's even kind of cooler, because it's chemistry, right? So if you're Christmas gift shopping, I suggest looking at scientific things. This is something you can actually buy, atomic seeds, irradiated with gamma radiation. So probably people see this and they think Incredible Hulk seeds, but you probably know better because you remember that the Incredible Hulk is probably just, most mutants are not the kind of mutants you see in comic books. Most mutants are bad, life is not easy to change that much. But you can see that these will have different results. You'll have one seed that's even one color, and sometimes you'll have disrupted the color-making gene, and the other seed will be completely bleached or albino, okay? So you can buy that. And there's just one last thing I want to see about how chemists can build machines like this. This is a chemist, James Tour. I've actually seen him speak. And he built this thing, which has this that he calls a stator, and this that has a rotor. Notice what's connecting the two, a double bond. And remember how, again, a theme in the class, double bonds can rotate. So you can imagine this can rotate 180 degrees. What he does is he builds different versions of this basic theme where you have different extensions off the end, different arms off the stator part. And what he did with these is he puts them on a membrane. And you notice how the stators, by the way, they're all kind of hydrophilic, but they're kind of hydrophobic too. It turns out that they like to interact with the top of the membrane. And then what they will do is they'll sit on the membrane, and then you start rotating the rotor. When you rotate the rotor, it starts to drill into the membrane. So you've made a molecular drill this way. Let me ask you, I don't think I've spilled the beans on this one. I said that you start to rotate the rotor, but I didn't say how. How would you rotate that double bond? Yeah, shine a light on it. So if you shine a light on it, it starts to rotate and it starts to form a pore. It starts to drill down into it. So yeah. So I think it's kind of cool, and I just want to say that James Tour is actually an outspoken Christian. He talks about his faith and his understanding. He's a very chemistry-oriented building stuff, so he's almost more like a physicist than a biochemist in my view. And actually, we have some disagreements, just to let you know. We have some disagreements about how biology and chemistry can interact and what possibilities are possible in biology. He's actually skeptical of origin of life from chemistry, which I'm very welcome to be skeptical. It's just I've become a lot less skeptical about that over the years, and now I think that it happened. He thinks it didn't. But it's great because we can be chemists and we can debate whether the chemistry can happen or not. So he has a talk about how he built little devices like this. He's famous for building a nano car, which looks like this. It's a little small in this depiction. It's 4 wheels of buckyballs, if you've heard of those. Those are carbon C60 arrangements. And they're on little molecular axles, and they actually roll along a surface. Okay? So he's literally building with these things. And yeah, you can put your chemistry knowledge to use in combining these types of molecules and putting it together. I do think that it's hard to be a professor. He submitted 37 proposals in 36 months, and most of those were as a single PI. And probably he got, if he was a above average science writer, that means he got 20% of those. That means like 7, right? So it takes a lot of writing of grant proposals to be a scientist. It actually takes a lot of mental strength and taking care of your own body, like the chess masters and things like that. But he was always there about how his family helped him when it's really hard to get a rejection letter, and when you're getting like, if you're only getting like 7, that means that 30 have been rejected, right? So that means that you have to actually have support. And he talks about how his family and his faith gave him the support to be a scientist that was producing at this level. Not all of us are called to make nanocars or to submit 36 proposals in 37 months or whatever it is. But I do like how he was supported in this work. This work is more than just chemistry to him. He actually needs others to be able to do this, even as the most brilliant scientist who puts these ideas together that I could never come up with. We all need each other, so I think that's an important thing to say. The very last thing to talk about is people acting like James Tour that are using 3D printing now to do some really amazing things. So 3D printing does not just have to work with like organic inks. You can use, as ink, you can use all sorts of things. Like for example, if you 3D print in oil, you can print a monolayer of a vesicle that will have a little aqueous bubble inside it. Then it'll have all of its chains sticking out. This is kind of like chocolate, actually, by the way. But you're printing little monolayer vesicles that will then stack up, and they'll form bilayers, but the bilayers will be membranes, okay? So you can actually print, and you can choose which nozzle it comes out of as you print. So you can choose to have it come out with red liquid in the middle or blue liquid in the middle. And so they show that you can do this, and they printed it out like this. Each of these, the pixels here, each of these is a little bubble of water held together inside a lipid. And you can see that the blue is not mixing with the orange. You can choose what you want to do, and this is about how big it looks in a human hand, by the way. So you can do it in a sphere, and you can make a stacked sphere, so you can make this little ball out of all of these things. You can make a little channel through the sphere of orange stuff, and you can make a three-dimensional channel. So you can think about this starts to look like vasculature or something like that. Because these are liquid inside, what you can do is you can actually come up with a way to put a pore into the membrane, and you can connect the different membranes this way. So what they did is they just made green ones, and they had hemolysin. Remember, hemolysin is just a big hole in the membrane. It's a protein that makes a hole. And so they have a situation like this where you have the hemolysin, the green ones are connected with pores. And they wanted to see, are they really connected? Well, let's put an electrode here, an electrode there, and they conduct electricity. You move the electrode up here, they don't conduct anymore. And so it shows that you've made sort of a little wire out of these little bubbles, a little bit like how nerves work. So that's another concept I want to bring back. So osmosis is actually can do cool things here. If you have high osmolarity next to low osmolarity, the one with high will swell, the one with low will shrink. If you have a whole stripe of those, then the whole stripe will curve in a particular direction. If you have a three-dimensional arrangement of those, the whole three-dimensional thing will curve up, and it will, when you move it to the different osmolarity, it will move from flat, and then it will curl up to make this kind of thing. Movie fans, sci-fi movie fans, does this remind you of anything? 1979, alien eggs, Okay, so I think that one of the reasons why those designs are so effective is because they are weird and lifelike. This is kind of like that, but much healthier. Okay. And so you can do it with collagen as well, instead of with vesicles. They actually do it at different pHs, and so you can say you put it out at pH 3.5, you can draw a line, and actually the surface will neutralize it, and so it will gradually neutralize as you go forward, and it will gel up. So they use this technology to print with collagen in the same way that they do that. And here's what they do. They print collagen walls, and then they can print with cells inside the collagen walls. So they can make a little cup like this, and it's cells inside collagen. It's a nice little 2-millimeter cup. That looks like a cardiac ventricle. They can do it with 3 valves, And that looks like a cardiac valve. Again, anatomy and physiology folks may be able to tell me how close this is, but it looks like my memory of a three-dimensional, of a tri-leaflet heart valve. And you can print a whole heart. This is what they did. So it's a 3D printing of collagen, and you can see that it's got these collagen walls. You can inject cells into it if you want, and anatomically, it is the right structure, and you can actually print out whatever structure you want because you can arrange the collagen however you want. Now, it's not fully functional yet, but they've got the valves, they've got it's the right consistency, it's the right size and shape, you know? I just think this is really cool, the idea of 3D printing. It sounds like Star Trek, you know? If you have a heart problem, could we eventually someday 3D print a new heart? Who knows? So I want to leave you with that, and I just want to, now let's talk about, that's an hour on all sorts of stuff that I want to talk about. Now, but what you really want to hear is how do you study for the test? And then there will be a little bit of time at the end for questions and stuff like that. So by the way, you should have this in your head, not on your body. Like I said, your head is part of your body, right? But this is, I think this is going too far. Although probably I'd just ask that you wear long sleeves if you do want to show your devotion to biochemistry by putting a molecule on. I'm trying to figure out which one that is. Is it sucrose? Yeah, that's right, because reducing ends, right? So there you go. There's a test question right there, you know. There's identical twins that have done sort of Fischer projection type things, and they've got the isomers of each other, you know, so the mirror images. So this D glucose and L glucose are shown right there. So there's a couple homework questions that I got. So one question that comes up sometimes is that when you see the model of the lipid, you will always see it drawn. You'll usually see it drawn as trans. But it's meant to be understood that yes, it can flip, and you might even have a little bit of trans in every sample. But usually it's going to mostly be in the low entropy spread out state, which means it will be in the cis state, if it is natural. Remember that cis is natural. If you know that, you can draw it however you want. So it's assumed that it's going to be cis unless you're told otherwise, if it's a natural lipid. That's the kind of thing for models, we just draw whatever's most convenient, and you have to interpret the model, which is why you can't just open a book and understand biochemistry. You have to build up the brain structures and the habits and the virtues, biochemical virtues. I'm talking like Lee Sanders, if you have ever taken a class from him. But that's that's why biochemistry takes so long. Okay. Any other sort of general quick questions like that before I go through the personality of the test? I looked through the sapling. I didn't see any that were giving. I saw that, like I expected, you haven't had that much time to do chapters 8 and 9. I didn't really see that much progress on chapters 8 and 9. I'll also look at it over the weekend. If there's a sapling problem that you have trouble with, one of the ways you can deal with it is just to have trouble with it, leave it incorrect, and go back to it later. I might be able to see that the whole class is having trouble with this problem and bring it up. Honestly, in this unit, though, they're more straightforward, so people do have less trouble, and I didn't see any evidence of that. But Peter. Yeah. Mm-hmm. Oh, yeah. Yeah, and that, okay. Yeah. So was the thing that tripped you up a little bit not having the sodium there, possibly? Yeah. And I understand for those drawing problems. I actually saw that one, and I saw that some people got that wrong. But then I looked at it, and the concept there, I'm pretty sure you've got, and it's just a question of whether what sapling wants for the details of where all of the bonds are. So, and in fact, honestly, if you just skip that one, it will be like, so if you have a drawing one and you're like, I've got this concept, it's probably in your interest to skip it, because I don't know how sapling's drawing interface is working these days. It used to be a pain, they made it better. I don't know how much better it is. So the drawing ones are, I'd like to give you credit for it, but if you need to skip it, I think if you have to skip something, skip those. But the concept there is base, breaks, esters, and it goes back to that particular thing. If you've got that concept down, that's what I want most of all. Yeah, but I did actually see that. So I will see it. I'll try to look, and maybe on Monday I'll see if I have, or you can actually, write me an email. That works too. Okay, so. All right, but let me go through the test, and if one of these jogs a question for you, go ahead and ask. There's going to be 150 points on the test. You notice that, as usual, there's an extra buffer of points. There's going to be 81 points of each of them. There's 27 multiple choice questions at 3 points each, covering all material from day one. I try to standardize these, and they're actually on a set number of topics. And so I don't let people take this test home, because it's not supposed to be studied like the other ones. If you happen to run across an old version of it, honestly, it's probably outdated anyways, but I'd like to ask on the honor system that you not look at it, just so I can have this be more consistent year to year. And that way I can see, this year, are they getting this concept, So this is the only one where I'm really going to ask that you don't look at previous tests if you can find them, and most people don't have them, get them back anyways, because it's Christmas. You don't want to test back on Christmas. Then there's a short answer part, and so the multiple choice is going to be all 4 units, and maybe a little more detail on unit 4, but not as much detail on units 1 to 3. But it's still going to be detailed, because it's the kind of thing, what kind of questions would they ask you possibly on the MCAT? That's kind of my, I want you to be ready for that, whether you're going to take it or not, So then there's short answer. There is half of it short answer, which will give you a chance to get partial credit and things like that. And completed sapling homework, as usual. One of the reasons why it's okay with me to skip one of the onerous drawing ones is because I'm always going to round up and add extra points on the sapling. So just try to do that. You'll get extra points for it if you can. As before, you'll need a calculator, although this test does probably have, it's test 3 that really has a lot of calculations on it. This one, I'm not quite sure yet exactly how it will turn out. There'll definitely be some calculations. Just don't be surprised if you are using it only on a few problems, though. Part 1 will be at a comprehensive level. It'll be like the ACS test, and it'll be like my version of the ACS test, so it might be a little harder, but it'll also help you if you go through Biochem 2. At the end of Biochem 2, you take the actual ACS test, and so I'm trying to prep you for that. Part 2 short answer will be at a unit test level, and that's where I'll give you a blank sheet of paper, ask you to draw sugar, things like that. That's where we have sort of the different level of recall. So yeah, this is the thing. I've never allowed students to take test 4 home. If you run across a copy, don't study it. Like I said, it's probably too old to be worthwhile of anything anyways. If you want to know what to study, look at the old tests and think about how would a question of this get at this general idea. Sometimes the old test questions are just more specific than a comprehensive test will ask. But they'll still be right and wrong, It's just that it won't be based on, like the readings. The readings are after general concepts, and you want to understand what those general concepts are. But I'm not going to go back to a reading and say, here, interpret figure 1, unless it illustrates a general concept really well and you can get it from the concept. I want you to be getting these things from the concepts at this point, not from remembering what was in the paper. And the MCAT's never going to ask you to have specific recall from one of the papers, but they are going to possibly ask you to interpret data, to understand at that level. And that's what I'm going for. Okay, so tips. Review the old tests primarily. Review old lectures, like skip through the videos and look at the headlines on the top. And for the ones where you're like, I'm still a little fuzzy on that concept, yeah, re-listen to those parts. But for the most part, I try to do things visually because, you don't need to go over everything, but you do need to have some kind of review to bring that stuff back, homeostasis, equilibrium. I try to have the top part be the most important concept. pH equations and pKa concepts, those actually come back because they're very universally applicable. They're applicable in pharmacy school, they're applicable for doctors using drugs and things like that. So pH and pKa and PI, those actually are important to even be able to work an equation. Henderson-Hasselbalch probably should remember that. I'm not sure, I probably won't give you a problem that's just work the Henderson-Hasselbalch. So if you have the general concept of pKa down, you might not need to use it. But if you are a learner who likes to have the equation in front of you, good news, you can memorize the equation, you can solve the problems that way. Okay. Of course, at this point, I expect to be able to say an amino acid and show it to you. I want you to do things like that glutenin peptide that I showed you, and to say to yourself, you know, which amino acid is most unusually enriched in this 20 residue thing. And you see the 3 prolines. So that was a little practice test question right there. Of course, I asked it in a much more vague kind of way because we're discussing, and there's multiple things that you could have said. So, but that's what I was going after ultimately. Biochem vocabulary are important if you have the same term with different kinds of ways. And you know, enzyme kinetics terms, to be able to be conversant with what's a KM. If I'm just sitting here and I'm talking to you about a KM, or if you're working at a drug company and they're talking about the KI or some things like that, they're not going to expect you to sit down and scribble out what the KI is. By the way, we said that that was too detailed already for test 3. But the concept of what big K means, and like the Ki is like a binding constant, so how do binding constants work? What kind of units do they use? How would you interpret data? Where is it on a graph? That's one thing you can always do. So biochem vocabulary includes words and pictures in the sense of a graph. And there will be, for at least the short answer part, I'm going to be giving you stuff from the literature. And so I want you to be able to put that together, and I promise, I know you haven't seen it before. I know that freaks you out, makes you burn calories, makes you breathe faster. Okay, so it's the chess grandmaster thing, and chess grandmasters have the same result, so don't feel bad about being stressed by a test, but realize I have given you a way out. And there's always something that you can take from the figure itself, the concept that I'm going after. And so I want you to be able to put together new ideas on the test. But it will never be a completely new idea, and I'll always give you the path through it. And then, if you're still stuck, just don't stay stuck. Just say to yourself, okay, that's the extra credit problem. I'll move on to the next one. Okay. So what to know? Carbohydrates, as mentioned in class, so that little lecture talks about them exactly. Fischer projections and Haworth perspectives, those are the 2 ways that we draw them. Be able to do that from monosaccharides. And the reason why I really want you to do this is I want you to be able to draw them in the little hexagon way, you know, the Haworth projection. But I want you to be able to read them in this way, which is related to the Haworth projection. I don't expect you to draw this well, but I want you to see this because everywhere in the literature, they talk about sugar structure using these. And if I'm copying and pasting from the literature, it's going to look like this. Realize that this is just a particular version of the Haworth projection. So you can definitely, you can look at this and you can say to yourself, oh, these are 2 hexasaccharides and they're flat, so it looks like it's glucose to me. And it looks like this one is alpha, opposite, and this one is beta, both on the same side. There's a little extra thing you have to do because they're showing you all of the carbons in true tetrahedral structure. But that's the only thing. There's all these terms we use, so I want you to be able to use the terms accurately. And then the differences between alpha and beta linkages, that's the difference between starch and cellulose. Okay? Optical rotation and mutarotation, we have those, and there's a homework problem about doing that. The homework problem, by the way, has a very specific equation. And of course, I don't want you to memorize the equation, but I want you to know how to use it. Like if I gave you a simple sort of concentration-based equation like that. It actually also connects to some of the stuff you do in lab. But more than anything, it connects to many problems in the homework. And you know, if you have a saturated lipid versus unsaturated, it's going to have different liquidity. It's going to have different melting point. How does that work? So types of lipids, we're going to have structures from figures mentioned in class. Notice that in some of those figures, some of those tables, I actually literally blocked out part of the table. And it's really about, there's a couple of more things to remember, like what's choline, okay? What is serine? It comes back, you know, so you already know that one, right? So know how to put those together. It's really only a couple of things to learn that are really new structures, but it's important to know how they go together. How fatty acids are converted to signaling molecules in general. We had hormones and prostaglandins. There are general categories. Of course, with signaling molecules, you've got tons of signals, okay? But you don't have to do that. You don't have to know the exact ones. You just have to know the general terms that apply to all of them. The fluid mosaic model and the bleaching experiment. One thing I like to do on the final exam is ask you to say back to me what you remember about a particular experiment. The good news is if you forget one of the 10 details, or 2, that's fine. You can still explain the experiment. You can just not mention that detail, okay? Be able to explain things back to me. Hydropathy plots, those show you where the helices that cross the membrane are. We showed you a couple examples of those. And a lot of the stuff about lipids is what parts of it are soluble, what's the effect of the different solubilities. Membranes, in a sense, are the effect of differentially soluble molecules. Fat-soluble vitamins, we had a few stories about those. Again, if I gave it a slide, pay attention to the slide, realize that the slide is fair game for some kind of question related to the general concept that it's getting after. And then we get to the last chapter, DNA and RNA structure and function. I've told you that I want you to draw the 5 possible nucleotides, be able to draw them in the base pairing form because that is such a fundamental insight. And if you are ever able to time travel back to 1951, you can actually take the credit from Watson because you can carry that idea back with you in your head and you can write the paper before Watson and Crick do. And you can give Rosalind Franklin credit. So if you're ever time traveling, I want to see a Doctor Who episode like that, but that's another story. It's a fundamental thing, 2 puzzle pieces fitting together to make base pairs. Be able to draw that. That has to do with the number of hydrogen bonds, have to do with DNA melting. Why we measure it at 260, whereas proteins we measure at 280. And then all of the transformations, those are non-enzymatic in the most part. We didn't talk about what enzymes do, we talked about what chemicals do. And so that's why I call it the chemical transformations. And then finally, sequencing and PCR. This is really at the level of being able to explain how it works. There's a tiny bit of organic chemistry with the phosphate group going on. And don't forget that. But realize that most of what's going on is reading off the complementary strand. The realization that DNA synthesis goes in the opposite direction of the template because that's the way that DNA strands work, you know. That's the biochemistry of it. There's a lot of homework problems on it. And this is a topic. Also, you might have this from other classes, and that would be a nice reinforcement, if so. So, like, don't know the optical rotation equation. Other minor equations are not important, like the delta G equations. The only delta G equation that is possibly important is the one that relates delta G to big K. Remember, equilibrium is a huge concept. Delta G equals negative RT natural log of K. But you've used that repeatedly, and it's actually a form of the same equation that we use for concentration and membrane crossing in Unit 3. So Henderson-Hasselbalch, the big K delta G equation, and I'm racking my brain. I don't want to accidentally miss something. There might be a third that's really important. Oh, Michaelis-Menten. I should have mentioned that. You use that so much that you should be able to do that, and that also holds several concepts together. There's lots of details, and I tried to mention these as I went through. This is not going to be important for the test. And weird bases beyond the 5 that we mentioned. Non-Watson-Crick base pairing. Oh, and I forgot, I didn't assign a reading this year, so definitely don't need to know about it. Okay? Sugars, be able to go with these, tip these over, turn them into the hexagon by tipping them to the right. Right goes down, left goes up. and you have some practice in your homework of doing that. Okay. And I already talked about this, actually, when we went through it. Just remember about that. The last thing I want to leave you is there is a secret Christmas message in sugar. And I got this from Alton Brown, so I need to cite it. If you actually take the sugar and you draw it this way, then it says ho, ho, ho all over them. So sugar goes with Santa, and as you're studying it, you at least have some practical application. You might even have practical usage of sucrose as you are studying. Please do online evaluations. Those are really important for me, and those help me from year to year. If 85% of the class does it, I promise you'll get at least 5 extra credit points. And if I've had a lot of sugar, I may even give a few more. So, okay. Yeah. And that's biochemistry. We're out of time right now, but if you have any questions for me right away, I'll also be around this afternoon. I'll try to have my door open. So I will see you on Tuesday at 10:30. Okay. What's up?
What is the preferred version of us giving you sugar?
Is this a bribery thing? That's against the Constitution, last I checked. Yes.
It's incentivization.
Yes. I have been known to accept all forms of sugar are pretty much good with me, except licorice. Yeah, so I'm not a licorice person. Yeah, well, it might have a negative effect. You have to realize there's such a thing as negative cooperativity. So.